Hello, everybody. How's everybody doing? Welcome to Chapter 10, from which we will cover blood in the human body. So, we are pretty much midway with anatomy and physiology. I hope everybody is taking their assessment examinations and doing quite well before moving forward to next chapters. Anyhow, before we begin, please remember to enable captions during the lecture, as additional terminology and reference points are provided there to support your understanding for physiologic concepts. So up to this point in this course, we have examined two major components of the cardiovascular system. First, we studied the heart, the muscular pump responsible for generating the force required to move blood through the body. Then we examined the blood vessels and hemodynamics, which had described the pathways and physical principles that regulate blood flow. So in this chapter, we will now focus on the third essential component of the circulatory system, which is blood itself. Blood is far more than a simple fluid circulating through the vessels. It is a highly specialized connective tissue that performs multiple life-sustaining functions. As mentioned from the previous chapter, blood is considered to be a connective tissue. So blood transports oxygen from the lungs to tissues, carries nutrients and hormones throughout the body. It also removes metabolic waste products, supports immune defense, and participates in hemostasis, which is the protective process that prevents excessive blood loss following an injury. So from a clinical perspective, understanding blood is critically important for safe healthcare practice. For example, blood transfusions are the most common medical interventions used to treat trauma, surgical blood loss, severe anemia, and other blood-related life-threatening or medical conditions. However, the incorrect administration of incompatible blood products can trigger acute hemolytic transfusion reactions. It is a dangerous immune response in which red blood cells are rapidly destroyed. So these reactions can lead to complications such as kidney failure, shock, or worst case scenario, even death. So because of this, healthcare professionals must understand the mechanisms of blood typing, antigen compatibility, and immune recognition to prevent additional or further serious clinical errors. So in this chapter, we will explore composition of blood, beginning with the liquid component known as the plasma, followed by formed elements which include erythrocytes, leukocytes, and platelets. We will also examine how blood cells are produced through hematopoiesis and how blood typing systems such as ABO and Rh groups determine transfusion compatibility. A clear understanding of how these principles is essential not only for mastering physiology, but also developing the clinical reasoning required in modern healthcare practice. As always, the goal of this course is to build a strong mechanistic foundation so that future clinical decisions are guided by accurate scientific understanding and, more importantly, patient safety principles. Blood is classified as a specialized connective tissue that circulates continuously through the cardiovascular system. Unlike most connective tissues, blood exists in a fluid state, allowing it to transport materials efficiently between organs and tissues. Despite its fluid appearance, blood is composed of two distinct structural components. The first component is plasma, which accounts for approximately 55% of total blood volume. Plasma is the liquid portion of blood and serves as the medium in which dissolved substances such as electrolytes, nutrients, hormones, proteins, and metabolic waste products are transported throughout the body. Suspended within the plasma are the formed elements, which make up roughly 45% of blood volume. These formed elements represent the cellular portion of blood and consist of three primary types. The most abundant are erythrocytes, also known as red blood cells, which specialize in the transport of oxygen and carbon dioxide. The second group consists of leukocytes, or white blood cells, which participate in immune defense by identifying and responding to pathogens or abnormal cells. The third component includes platelets, also called thrombocytes, which are small cellular fragments that play a central role in hemostasis, the process that prevents excessive bleeding following vascular injury. The relative proportion of formed elements within blood is commonly measured using a laboratory parameter known as hematocrit, which represents the percentage of blood volume occupied by red blood cells. Hematocrit values are clinically important because they help healthcare professionals assess conditions such as anemia, dehydration, or blood loss. Understanding the basic division between plasma and formed elements provide the structural framework for the remainder of this chapter. Each component contributes uniquely to essential functions of blood, including transport, immune protection, and hemostatic balance. Laboratory evaluation of hematocrit and blood cell counts is commonly used to diagnose conditions such as anemia, infection, dehydration, and internal bleeding. Misinterpretation of hematocrit values or blood cell counts can delay recognition of serious clinical conditions such as hemorrhage, sepsis, or severe anemia. After understanding that blood is composed of plasma and formed elements, we now begin examining the largest component of blood, which is plasma. Plasma is the liquid matrix of blood and accounts for approximately 55% of total blood volume. It appears as pale yellow or straw-colored fluid and serves as the medium in which the cellular component of blood are suspended and transported throughout the circulatory system. The majority of plasma is composed of water, making up roughly 90% of its total composition. The high water content allows plasma to function as an efficient transport medium, carrying dissolved substances throughout the body. Within this fluid environment, plasma transports electrolytes, nutrients, hormones, metabolic waste products, and various proteins that are essential for maintaining physiologic balance. Among the most important components dissolved in plasma are plasma proteins, which include albumin, globulins, and fibrinogen. These proteins perform several critical functions. Albumin helps maintain osmotic pressure, which prevents excessive fluid loss from the bloodstream into surrounding tissues. Globulins participate in immune defense, and fibrinogen plays an essential role in blood clot formation. So from a clinical perspective, plasma composition is routinely evaluated through laboratory testing, Measurements of electrolytes, plasma proteins, and metabolic waste products provide healthcare professionals with valuable information about the patient's hydration status, kidney function, liver function, and overall metabolic balance. Understanding plasma as the transport medium of the circulatory system provides an important foundation for interpreting many laboratory values that nurses encounter in clinical practice. Laboratory analysis of plasma electrolytes, proteins, and metabolic waste products provides critical information for assessing hydration status, kidney function, liver function, and metabolic balance. Although plasma is composed mostly of water, its most important dissolved components are plasma proteins. These proteins play essential roles in maintaining the stability and proper function of the circulatory system. The most abundant plasma protein is albumin, which is responsible for maintaining colloid osmotic pressure within the bloodstream. This osmotic pressure helps keep fluid within blood vessels and prevents excessive fluid leakage into the surrounding tissues. When albumin levels decrease significantly, fluid may accumulate in tissues leading to edema. The second group of plasma proteins are globulins. Many globulins function as antibodies, which are part of the immune system and help the body recognize and neutralize pathogens such as bacteria and viruses. The third major plasma protein is fibrinogen, which plays a central role in blood clot formation. During vascular injury, fibrinogen is converted into fibrin, forming a network that stabilizes blood clots and prevents excessive blood loss. From a clinical perspective, plasma protein levels are important indicators of patient health. For abnormal or abnormal albumin levels may indicate liver disease, malnutrition, or kidney disorders, while abnormalities in clotting proteins may increase the risk of bleeding complications. So understanding the functions of plasma proteins provide important insight into how the circulatory system maintains fluid balance, immune protection, and hemostasis. Clinically, low albumin levels may lead to edema, while abnormalities in clotting proteins can increase the risk of bleeding disorders. In addition to plasma, the second major component of blood consists of formed elements, which represent the cellular portion of blood. These components make up approximately 45% of total blood volume and are responsible for many of the critical physiologic functions of the circulatory system. There are three major types of formed elements. The first and most abundant are erythrocytes, also known as red blood cells. These cells specialize in the transport of oxygen and carbon dioxide between the lungs and body tissues. The second group consists of leukocytes, or white blood cells. Leukocytes play an essential role in immune defense, protecting the body from pathogens such as bacteria, viruses, and other foreign substances. The third group includes platelets, also called thrombocytes. Platelets are small cellular fragments that are responsible for initiating blood clot formation, which helps prevent excessive bleeding following injury to blood vessels. So together, these three components allow blood to form several vital functions. Erythrocytes support oxygen delivery, leukocytes protect against infection, and platelets maintain hemostasis by preventing blood loss. In clinical practice, changes in the number of function of these formed elements can indicate important medical conditions. For example, decreased erythrocytes may suggest anemia, elevated leukocytes may indicate infection, and reduced platelet counts can increase the risk of bleeding disorders. Understanding these formed elements provides the foundation for interpreting many common laboratory tests, particularly the complete blood count or CBC, which evaluates the cellular composition of blood. The complete blood count or CBC evaluates the number of red blood cells, white blood cells, and platelets, helping clinicians detect conditions such as anemia, infection, and bleeding disorders. Among the formed elements of blood, the most numerous cells are erythrocytes, also known as red blood cells. These cells are specialized for the transport of oxygen throughout the body. Erythrocytes have distinctive biconcave disc shape, meaning they are thinner in the center and thicker around the edges. This shape increases the surface area available for gas exchange, allowing oxygen to diffuse efficiently between the lungs and circulating blood. Unlike most other cells in the body, mature erythrocytes lack a nucleus and most organelles. This structural adaptation allows more internal space to be devoted to protein hemoglobin, which is responsible for carrying oxygen molecules. Each hemoglobin molecule contains iron atoms that bind oxygen in the lungs and release it to body tissues where oxygen is needed for cellular metabolism. Through this mechanism, erythrocytes enable the bloodstream to deliver oxygen efficiently to organs and tissues throughout the body. From a clinical perspective, abnormalities in erythrocyte number or hemoglobin concentration can lead to conditions such as anemia, which reduces the blood's ability to transport oxygen. Symptoms of anemia may include fatigue, shortness of breath, and decreased exercise tolerance. For this reason, laboratory tests such as hemoglobin levels and hematocrit measurements are important indicators of a patient's oxygen carrying capacity. From a clinical perspective, a low hemoglobin or erythrocyte levels may indicate anemia, reducing the blood's ability to deliver oxygen to tissues. The ability of erythrocytes to transport oxygen depends primarily on a specialized protein known as hemoglobin. Hemoglobin is an iron-containing protein located inside red blood cells. Each hemoglobin molecule has the ability to bind oxygen molecules, allowing erythrocytes to carry oxygen efficiently through the bloodstream. In the lungs, oxygen enters the bloodstream through the pulmonary capillaries. At this location, oxygen molecules bind to hemoglobin within the erythrocytes. Once oxygen is attached, the blood carries it through the systemic circulation, delivering oxygen to tissues throughout the body. When erythrocytes reach tissues where oxygen is needed for cellular metabolism, hemoglobin releases the oxygen molecules. These oxygen molecules then diffuse into surrounding cells where they support the production of energy through cellular respiration. Because hemoglobin plays such an essential role in oxygen delivery, its concentration in the blood is a critical clinical measurement. Low hemoglobin levels reduce the blood's ability to transport oxygen and may lead to symptoms such as fatigue, weakness, and shortness of breath. So for this reason, hemoglobin levels are routinely measured as part of a complete blood count, helping healthcare providers evaluate conditions such as anemia, blood loss, or chronic disease. So therefore, a low hemoglobin levels or low hemoglobin levels reduce oxygen delivery to tissues and are key indicators of anemia. In addition to transporting oxygen, blood also plays a crucial role in protecting the body against infection. This protective function is carried out by leukocytes, commonly known as white blood cells. Leukocytes are specialized cells that identify, attack, and remove pathogens. These include bacteria, viruses, and other foreign substances that may enter the body. Although they are far fewer in number than erythrocytes, their role in maintaining health is essential and important. There are several different types of leukocytes, each with a specific function in the immune system. We have first the neutrophils, the most abundant type, and are particularly effective at destroying bacteria. Next, we have lymphocytes. They are responsible for coordinating immune response and producing antibodies. Next, we have monocytes. They develop into macrophages that engulf and digest pathogens through a process known as phagocytosis. Two additional leukocyte types are eosinophils, which are involved in defense against parasites and allergic reactions, and basophils, which release chemical mediators that contribute to inflammation. In clinical practice, the number of leukocytes in the bloodstream is measured through a white blood cell count, which is part of the complete blood count test or CBC test. Elevated blood cells levels may indicate infection, inflammation, or immune activation, while abnormally low levels may suggest immune suppression or bone marrow disorders. For example, common among patients with leukemia disorders. So understanding the function of leukocytes helps healthcare professionals interpret laboratory results and identify signs of infection or immune dysfunction. CBC results with elevated blood cell counts may indicate infection or inflammation, while decreased counts may suggest immune suppression or bone marrow dysfunction, such as patients suffering from leukemia. In addition to erythrocytes and leukocytes, blood contains a third type of formed element known as platelets, also called thrombocytes. Unlike red and white blood cells, platelets are not complete cells. Instead, they are small cellular fragments derived from large bone marrow cells known as megakaryocytes. These fragments circulate within the bloodstream and play a crucial role in preventing excessive blood loss. When a blood vessel becomes damaged, platelets are among the first components of blood to respond. They rapidly adhere to exposed surface of the injured vessel wall and begin to stick to one another in a process known as platelet aggregation. This aggregation forms an initial platelet plug, which temporarily seals the damaged vessel. At the same time, a series of chemical reactions known as the coagulation cascade activates clotting proteins in the plasma. These proteins form fibrin strands that strengthens and stabilize the developing clot. Together, these processes are referred to as hemostasis, which is the body's mechanisms for stopping bleeding and maintaining vascular integrity. So from a clinical perspective, platelet counts are an important laboratory measurement. Low platelet levels, a condition known as thrombocytopenia, increase the risk of bleeding, Conversely, excessive platelet activity may contribute to abnormal clot formation, which can lead to serious complications such as stroke or thrombosis. Understanding platelet function helps healthcare professionals evaluate bleeding risk and interpret laboratory findings related to clotting disorders. Clinically, low platelet counts or thrombocytopenia increase the risk of excessive bleeding, while abnormal clot formation may contribute to thrombosis or stroke disorders. All of the formed elements of blood originate from a common process known as hematopoiesis, which refers to the production and development of blood vessels. Hematopoiesis occur primarily within the red bone marrow, a specialized tissue found in certain bones of the body, including the sternum, ribs, pelvis, and vertebrae, and the long femur. Within this environment, immature precursor cells known as hematopoietic stem cells divide and differentiate into the various types of blood cells. Through this process, the bone marrow continuously produces erythrocytes, leukocytes, and platelets, ensuring that circulatory system maintain adequate oxygen transport, immune protection, and clotting capability. One of the most important regulators of blood cell production is a hormone known as erythropoietin, often abbreviated as EPO. This hormone is produced primarily by the kidneys in response to low oxygen levels in the blood. When oxygen level decreases, erythropoietin stimulates the bone marrow to increase the production of erythrocytes, thereby improving the body's oxygen carrying capacity. In clinical practice, disorders affecting the bone marrow can disrupt normal hematopoiesis. Conditions such as bone marrow failure, leukemia, or certain chemotherapy treatments may reduce the production of blood cells, leading to anemia, risk of infection, or impaired clotting. Understanding hematopoiesis helps healthcare professionals interpret laboratory findings and recognize diseases that affect blood cell production. For example, disorders of bone marrow function can reduce blood cell production, leading to anemia, risk of infection, or bleeding disorders. The ABO blood group system is the most important classification used to determine red blood cell transfusion compatibility. For healthcare professionals, this knowledge is not optional because an incorrect blood transfusion can produce severe and life-threatening reactions. Blood types are determined by the presence or absence of specific antigens located on the surface of erythrocytes or red blood cells. These antigens act as identifying markers that the immune system can recognize. Individuals with type A blood have A antigens on their erythrocytes. Individuals with type B blood have B antigens. Individuals with type AB blood possess both A and B antigens, while individuals with type O blood have neither A nor B antigens on the surface of their red blood cells. In addition to these antigens, the plasma contains antibodies that react against foreign antigens. For example, individuals with type A blood produce anti-B bodies or anti-B antibodies, while individuals with type B blood produce anti-A antibodies. Individuals with type AB blood do not produce anti-A or anti-B antibodies, while individuals with type O blood produce both anti-A and anti-B antibodies. These antigen-antibody relationship determine which blood types can be safely received during a transfusion. If a patient receives blood containing antigens that react with antibodies in their plasma, the red blood cells will clump together in a process known as agglutination. This reaction can destroy red blood cells and may result in hemolytic transfusion reactions, which can lead to shock, kidney failure, or even death. The compatibility table shown on this slide summarizes the critical relationships between blood type, antigens, antibodies, and transfusion compatibility. It identifies which blood types a patient can safely receive, which blood types they can donate, and which blood types must never be transfused due to antibody reactions. Two important clinical principles help simplify transfusion compatibility. Type O blood is considered the universal red blood cell donor because it lacks both A and B antigens. Type AB blood is considered the universal red blood cell recipient because it does not contain antibodies that attack A or B antigens. Because transfusion errors can have fatal consequences, healthcare professionals must always verify patient identity, blood type, and compatibility testing before administering any blood product. In addition to the ABO blood group system, another important blood classification used in transfusion medicine is the Rh factor. The Rh factor is determined by the presence or absence of the Rhd antigen located on the surface of erythrocytes. Individuals who possess this antigen are classified as Rh positive, while individuals who lack the antigen are classified as Rh negative. The Rh factor is important in two major clinical situations: blood transfusions and pregnancy. So during blood transfusions, compatibility must be verified to prevent immune reactions. Rh positive patients can receive red blood cells from either Rh positive or Rh negative donors because their immune system already recognizes the Rh antigen. However, Rh negative patients should receive only Rh negative blood whenever possible. If an Rh negative individual receives Rh blood, their immune system may recognize the Rh antigen as foreign and begin producing anti-Rh antibodies. Once these antibodies develop, future exposure to Rh positive blood can trigger hemolytic transfusion reactions, which may destroy red blood cells and cause serious complications. To illustrate this clinically, consider a patient whose blood type is A positive or A plus as normally labeled on real-life blood products. This means that the patient has A antigens and the Rh antigen on their erythrocytes. Because of this, A plus patient or A positive patient may safely receive red blood cells from A plus or A positive, A negative, O positive, or O negative donors, since type O blood is considered a universal donor blood. By contrast, a patient with A negative blood does not possess the Rh antigen. This patient should ideally receive A negative or O negative blood type only, because exposure to Rh positive could stimulate the immune system to produce anti-Rh antibodies. The Rh factor is also critically important during pregnancy. If an Rh negative mother carries an Rh positive fetus, fetal red blood cells may enter the maternal circulation during pregnancy or delivery. This exposure can stimulate the mother's immune system to produce anti-Rh antibodies. During a subsequent pregnancy with another Rh positive fetus, these maternal antibodies may cross the placenta and attack fetal red blood cells, a condition known as hemolytic disease of the newborn, or also called erythroblastosis fetalis. Fortunately, this condition can be prevented through the administration of Rhod immune globulin, commonly known as RhoGAM, which prevents the mother's immune system from developing antibodies against Rh positive blood cells. Because of these risks, healthcare professionals must always verify ABO type, Rh status, and cross-match testing before administering blood products. One of the most frequently ordered laboratory tests in clinical practice is the complete blood count, or CBC. This evaluates the major cellular components of blood, including red blood cells, white blood cells, and platelets, and provides important information about a patient's physiological status. Several key measurements are included in a CBC. One of the most important is hemoglobin, often abbreviated as HGB. Hemoglobin is the oxygen-carrying protein found within the erythrocytes. When hemoglobin levels are low, the blood's ability to transport oxygen is reduced, which may lead to symptoms such as fatigue, weakness, and shortness of breath. In this example, the hemoglobin value is about 10.5 grams per deciliter, which is below the normal range and may indicate anemia. Another important value is the hematocrit, abbreviated HCT. Hematocrit represents the percentage of blood volume composed of red blood cells. Abnormal hematocrit values may suggest conditions such as blood loss or dehydration, depending on whether the value is decreased or elevated. Notice how the abnormal values in the report correspond directly to the clinical meaning shown in the right column. The CBC also measures the white blood cell count, or WBC, which reflects immune system activity. Elevated white blood cells level may occur during infection or inflammation as the body responds to invading pathogens. Another critical component is platelet count, which reflects the blood's ability to form clots. Platelets play an essential role in hemostasis, the process that stops bleeding after vascular injury. Abnormally low platelet levels may increase the risk of bleeding disorders, while excessively high levels may increase the risk of abnormal clot formation. Normal CBC values vary slightly between females and males, particularly for measurements related to red blood cells. For example, normal hemoglobin levels are typically about 12 to 16 grams per deciliter in females and approximately 13.5 to 17.5 grams per deciliter in males. Similarly, hematocrit values are usually around 36 to 46 in female and about 40 to 52% in males. These differences are largely influenced by hormonal effects and red blood cell production. So by interpreting CBC values together, healthcare professionals can identify potential health concerns such as anemia, infection, inflammation, bleeding disorders, or bone marrow abnormalities. Abnormal CBC findings therefore require further evaluation and appropriate clinical management. Abnormalities in blood cell counts may indicate a variety of underlying medical conditions. These disorders typically affect red blood cells, white blood cells, or platelets, and are often detected through laboratory tests such as the complete blood count or CBC. One of the most common blood disorders is anemia, which occurs when hemoglobin levels or red blood cell counts fall below normal levels. Anemia reduces the blood's ability to transport oxygen, often carrying symptoms such as fatigue, weakness, dizziness, and even shortness of breath. In contrast, polycythemia occurs when body produces an excessive number of red blood cells. This increases the blood viscosity, meaning the blood becomes thicker and may increase the risk of blood clots, stroke, or cardiovascular complications. Disorders can also affect white blood cells. Leukocytosis refers to an elevated white blood cell count, which commonly occurs during infection or inflammation as the immune system responds to pathogens. Conversely, leukopenia describes an abnormally low white blood cell count. This condition may occur due to bone marrow suppression, certain medications, viral infections, or chemotherapy, and it increases the patient's risk of infection. Platelet disorders also have an important clinical consequences. Thrombocytopenia occurs when platelet levels are too low, which increases the risk of excessive bleeding or bruising because the blood cannot clot properly. Now, on the other hand, thrombocytosis occurs when platelet levels are elevated. This may increase the risk of abnormal clot formation, which can lead to conditions such as deep vein thrombosis or DVT, pulmonary embolism or PE. Recognizing these abnormalities allows healthcare professionals to identify potential diseases, disease processes, and guide further diagnostic testing and treatment. In this chapter, we examine the structure, composition, and functions of blood, an essential connective tissue responsible for transport, immune defense, and regulation of physiological stability. Blood consists of two primary components, plasma and formed elements. Plasma serves as the liquid matrix that transports proteins, nutrients, hormones, electrolytes, and metabolic waste throughout the body. The formed elements of blood includes erythrocytes, leukocytes, and platelets. Erythrocytes contain hemoglobin and are responsible for transporting oxygen to tissues. Leukocytes function as part of the immune system and help protect the body against infection. Platelets play a critical role in hemostasis, allowing blood to clot following vascular injury. We also explored blood typing systems, including the ABO blood group system and the Rh factor, both of which are essential for determining safe blood transfusion compatibility. Incorrect transfusion of incompatible blood may result in hemolytic transfusion reaction, which can have life-threatening consequences. Finally, we discuss how the complete blood count, or CBC, provides valuable diagnostic information by evaluating red blood cells, white blood cells, and platelets. Abnormal CBC values may indicate conditions such as anemia, infection, or bleeding disorders requiring further clinical evaluation. Understanding the composition and function of blood is essential for recognizing disease processes and ensuring safe clinical practice. Before proceeding to the next chapter, learners should complete the chapter assessments to demonstrate mastery of these concepts. And a reminder from SSRP, in healthcare, knowledge is not optional and errors carry real life consequences.